For the first time in human history, we have a tool that can directly edit DNA. It allows scientists to fix genetic mistakes, treat certain diseases, and potentially prevent them from being passed on. This isn't a distant future or a theoretical idea. This technology is already being used in real humans, in crops, and in animals. It's called CRISPR. And in this video, we'll explore what gene editing really means and why it matters. So, what exactly is CRISPR? CRISPR is a biological tool, a tool that comes from nature. Long before humans ever thought about editing DNA, bacteria were already doing it, not to cure diseases, but to survive. Bacteria are constantly attacked by viruses, and over millions of years, they evolved a defense system. When a virus attacks, bacteria capture small pieces of the virus's DNA and store them inside their own genome, like a biological memory. If that same virus attacks again, the bacteria recognize it immediately, cut the virus's DNA, and stop the infection before it spreads. That natural defense system is called CRISPR. At its core, CRISPR is a memory system combined with a precision defense mechanism. And this is the moment everything changed. Scientists realized that if nature already knows how to recognize specific DNA and cut it, maybe we can guide that system to cut any DNA we choose. Today, when we talk about CRISPR, we're usually talking about a system called CRISPR-Cas9. Cas9 is a protein that acts like molecular scissors. CRISPR acts like a guide, telling those scissors exactly where to cut. Together, they allow scientists to enter the DNA of a living cell, locate a precise genetic sequence, and edit it. That edit could mean removing a harmful gene, fixing a broken one, or replacing it with something new. Every cell in your body follows DNA instructions. Those instructions decide how a cell functions, how it repairs itself, how it grows, and sometimes how it fails. When those instructions contained an error, we had no way to fix them. All we could do was manage the consequences. CRISPR changes that. For the first time, scientists can go directly into the genetic code of a living cell and interact with the instructions themselves. They can locate a faulty instruction, they can remove it, repair it, or replace it with a corrected version. This is not an upgrade to traditional medicine. It is a completely different approach. Most medicines work around a problem. They reduce symptoms, slow damage, or support the body. CRISPR works at the source of the problem. If a disease is caused by a single genetic error, CRISPR can target that exact sequence, and only that sequence. And once that correction is made, every new cell created from that DNA carries the fix forward. Instead of managing symptoms, CRISPR allows us to rewrite the instructions themselves. The goal of CRISPR is not to create something new. It's to correct what went wrong. Many of the most serious diseases humans face don't begin with an infection or an injury. They begin much earlier, inside the DNA itself. A single typo in the genetic code can disrupt how cells behave, divide, or survive. That one error can lead to lifelong conditions, inherited disorders, or diseases that appear later in life. And if you had children, there was a chance that the same genetic error would be passed on again. CRISPR introduces a different goal. Instead of responding to the consequences of genetic errors, the goal is to intervene at the origin and correct it directly. If harmful genetic changes can be corrected early enough, they don't just stop affecting one person, they stop being passed on. That shift, from accepting genetic fate to actively correcting it, is what makes CRISPR different from anything before it. In the late 1980s and early 1990s, scientists studying bacteria noticed something strange in their DNA. Short, repeating sequences that didn't seem to serve any obvious purpose. At first, no one knew what they were. Years later, researchers realized those repeats were not random. They were fragments of viral DNA. Bacteria were keeping records of past infections. In the early 2000s, scientists finally understood what was happening. Bacteria were using those stored sequences as a defense system, a way to recognize and destroy viruses they had encountered before. That system was named CRISPR. But the real turning point came in 2012. That's when researchers demonstrated that CRISPR, combined with a protein called Cas9, could be programmed, not just to defend bacteria, but to cut any DNA sequence scientists chose. Since then, CRISPR has rapidly evolved, more precise versions, more efficient systems, new proteins beyond Cas9. Each improvement made gene editing faster, cheaper, and more accurate. What comes next with CRISPR is a range of possibilities. Right now, CRISPR is still mostly used in controlled environments, specific diseases, specific cells, specific cases, but the direction is clear. As this technology becomes more precise, more affordable, and more regulated, its impact will quietly expand. Outside of humans, its influence is already spreading. In agriculture, CRISPR is being used to create crops that survive droughts, resist disease, and grow with fewer resources. But with that power comes responsibility, because the same tool that can correct harmful mutations also forces us to decide where the line is, not about what can be done, but about what should be done. And those decisions won't be made by scientists alone. They'll be shaped by laws, ethics, culture, and public trust. The benefits of CRISPR go far beyond curing disease. If we understand genes well enough, CRISPR could eventually help enhance how bodies function, stronger resistance to disease, improved recovery, better tolerance to extreme environments. CRISPR gives us a level of power humanity has never held before. And like every powerful technology before it, the question is not can we use it, but how far should we use it? CRISPR can heal. It can remove suffering. It can prevent diseases that have shaped human lives for generations. But once we change our genetics, there is no undo button. Those changes don't just affect one person. They can carry on for generations. That's why law, regulation, and restraint matter as much as science itself. Not to stop progress, but to guide it. The coming decades will decide where we draw the line between healing and enhancement, between correction and control, and how we choose to use it will shape what it means to be human for a very long time.